Neo7 Bioscience and McCullough Foundation presents Autism involves mitochondria. Nuclear regulatory failure, not primary mtDNA damage, drives dysfunction. Mitochondrial dysfunction is one of the most consistently observed biological abnormalities in autism spectrum disorder. That observation is correct. The interpretation, however, has been persistently flawed. Let me state this clearly at the outset. Mitochondria are involved in autism. Autism is not, in the overwhelming majority of cases, a mitochondrial DNA inheritance disease. The failure occurs upstream within nuclear regulatory systems that govern mitochondrial behavior, immune signaling, and metabolic control. In rigorously characterized cohorts of children with autism and mitochondrial dysfunction, fewer than 16% demonstrate detectable mitochondrial DNA mutations, deletions, or depletion. More than 84% show intact mitochondrial DNA despite clear dysfunction. This leads to a firm conclusion: mitochondrial dysfunction in autism is real, but it is overwhelmingly not caused by inherited mitochondrial DNA failure. The mitochondrial genome contains just 37 genes. The nuclear genome encodes more than 1,100 mitochondrial-associated proteins. Less than 1% of mitochondrial control is derived from mitochondrial DNA. More than 99% is nuclear-controlled. What we observe in autism is not mitochondrial collapse. It is regulatory failure. In autism, mitochondria are often structurally preserved but functionally mismanaged. We see inefficient energy production, redox imbalance, abnormal stress signaling, and poor coordination between neuronal demand and energy supply. What we do not see in most cases are progressive mitochondrial encephalopathy, high-burden heteroplasmy, or classic maternal inheritance patterns. The mitochondria are responding to corrupted instructions. Nuclear regulatory systems are epigenetically vulnerable. Environmental toxicants and non-precision immune stressors can disrupt transcriptional control, destabilize calcium and immune signaling, and lock cells into chronic stress states. This does not require mitochondrial DNA damage. It requires nuclear regulatory vulnerability. This is why autism cannot be addressed with generic mitochondrial supplements or one-size-fits-all immune interventions. You cannot correct a control system failure by ignoring the control layer. At Neo7 Bioscience, precision systems biology focuses on stabilizing disrupted nuclear regulatory networks, supporting mitochondrial governance, and restoring immune-metabolic coordination. This work aligns with the McCullough Foundation's commitment to scientific integrity, biological truth, and patient-centered investigation. Let this be stated without ambiguity. Mitochondria are involved in autism. Primary mitochondrial DNA disease is uncommon. The overwhelming majority of dysfunction reflects nuclear regulatory failure. The mitochondria are not broken. They are mismanaged. Autism is a disorder of biological governance, not genetic inevitability. Precision biology must come first. At Neo7 Bioscience, personal molecular surveillance and personalized engineering directly target deficient biological signals. Matched to each individual's unique expression to correct signal flaws, deficiencies, and regulatory impairments at their source.